Rita is a distinguished University of Maryland professor. She also is the 11th director of the National Science Foundation. She's one of the world's foremost experts in Vibrio, a dangerous bacteria that has the potential to contaminate seafood and drinking water, leading to foodborne infection and other serious adverse effects. Her accomplishments have earned her the National Medal of Science, awarded by President George W. Bush, the International Prize for Biology, the Japan Marine Biology Prize from the Emperor of Japan, the Stockholm Water Prize awarded by the King of Sweden for basic research, and the Singapore Water Prize for her applied work, of which she was the first female double winner. She received her BS in bacteriology and an MS in genetics, both from Purdue University. She was awarded her PhD from the University of Washington in the field of aquatic microbiology or oceanography. She's been awarded 62 honorary degrees from institutions of higher education, including her alma mater, Purdue University. She was inducted into the National Women's Hall of Fame and has a new book on the way about women in science. She truly believes that water is one of the biggest challenges facing humanity today, becoming a trade-off for drinking, domestic use, agricultural application, industry, and more. It's not just knowing how much and where, but how to allocate usage of water and how to share with competing priorities. Please help me welcome Rita Colwell to the podium.
Thank you very much for the very kind introduction. It's an honor to be able to speak to you, and I'm very pleased to have been able to see the poster sessions and to meet some of the students and faculty. It's great that these are the good things that are happening right here at the University of Maryland. And what I'm going to do is speak about waterborne diseases and their transmission, as I've been working on cholera all of my career. And the results that I'm going to be presenting were all obtained right here at the University of Maryland. So first of all, let me just point out that water-related diseases are major killers, and diarrheal disease, including cholera, is the worst of all. And you read about the cholera epidemics in Haiti and the current epidemics in Yemen, and I'll tell you a bit about that, those epidemics as I proceed in my talk. But the point is, when you add up all of the deaths and the cases per year, it gets to be the point of billions. And those who do not have access to safe water number something in the order of 2 or 3 billion. Cholera is a global disease. It's a water-related disease. It is numbered, or the numbers of cases are in a series of pandemics. We're in what's called the 7th pandemic right now, with the outbreaks in Haiti, Yemen, and in Asia. More than 50 countries are affected, about 7 million people. And predominantly in Bangladesh, cholera exists in the aquatic habitat. I'll be speaking about that in a moment. It's very unlikely that we can eradicate cholera, but it clearly can be controlled. For example, I think in this room, very few of you have ever had the cholera vaccine. Some of us had, who've worked in cholera endemic countries. But frankly, because we are lucky to have safe, treated drinking water, we don't have to worry about the disease. Now, in the old days, back 100 years ago, it was considered a port of entry disease. In other words, ships coming in and in the port area, the disease would occur. This is from the New York Times of about 1875, saying science is asleep while cholera comes into New York. It's easily cultured, and it's a little bacterium with a polar flagellum. The disease itself, though, is devastating. You crudely measure the fluid loss, but if not rehydrated, usually by IV or treated with antibiotics and fluid replacement, death can occur, and quite quickly. If you read any of Rudyard Kipling's novels, or had done, you'll know that the saying was, fine at breakfast, dead at dinner. So it can occur very quickly. Now, here at the University of Maryland, when I first came to Maryland, we did a lot of research up and down the bay. This is a very familiar chart. And these are the stations that we sampled quite regularly. And we were lucky to use the Horn Point Laboratory, the Patuxent Laboratory as well. So the research that we carried out led to some discoveries. In 1975, we were able to publish a paper showing that Vibrios are associated with Copepods, and there is an annual cycle, particularly toward the end of the year in the Chesapeake Bay, when the bloom of Copepods crashes and then releases the Vibrios into the water column. So the association, is it still working? No, it's not. Okay. I guess I'll use the mic. The association with Copepods was controversial because it was believed that cholera can only be transmitted person to person. And this is the critter that's familiar to those of you who work in the Chesapeake Bay, the Calanus copepod host of Vibrios and including Vibrio cholerae. Now, we developed a very crude model at that time. This was in 1975, 1980, where essentially we felt that transmission occurred because first you would have a bloom in the spring of the, let me turn this off. You'd have a bloom in the spring when sunlight becomes more abundant, and the phytoplankton, that is those that carry chlorophyll, become very abundant. The zooplankton then feed on the phytoplankton, and when the crash of that is the reduction in nutrient and the drop in the population, the breakup of the copepods leads to Vibrios being freely swimming in the water column. And if the water is drunk that has the bacteria in it, then as a result, there'll be cases of cholera. Now in Maryland, we had cholera until about until 1920. So it was a disease. In fact, if any of you hiked the C&O Canal, the Chesapeake and Ohio Canal, there is on one of the overpasses, there is a concrete monument to the canal workers who died of cholera building the C&O Canal. Now, the interesting other observation that we made, it's really very fundamental, that is in aquatic systems, we were able to show that if you used a staining technique, a fluorescent antibody, that is an antibody linked to a fluorescent molecule, and then viewed under an epifluorescent microscope, it glows. And you can count the bacteria very nicely. And the counts were always much higher than what we could plate, that is, what we could culture, the yellow columns. And so that led us to the hypothesis that, in fact, there were bacteria that went into a dormant stage. This was 1980 at the University of Maryland, and it was not well received. The fact that you couldn't culture them, they had to be dead. Of course, we now know with DNA techniques that by far 98% of the microorganisms still have not been cultured. But that was a major finding here at the University of Maryland. And we developed a technique to be able to see them by using the antibody that was linked to a fluorescent molecule. And we also showed with electron microscopy that the bacteria go into a kind of spore-like stage. Now, these are gram-negative bacteria, meaning they don't have a spore coat. They can't form one. But because they're aquatic, they really don't need to. So they can become dormant without having formed a hard shell to be protected. If you're living in soil, like bacillus and those species, you have to form a spore coat to avoid desiccation. So that was another major discovery made here at the University of Maryland. And we developed a technique for rapid diagnostics by using that antibody and a little device, a kind of a clamshell device where you could put the sample from a stool or from water or whatever, and then it would turn a color with a reagent that was present, and you could do a diagnostics or diagnose cholera within a minute or two. And this was important in the field. Now, cholera as a waterborne disease is global. The darker color is the more intense the epidemics that occur. And it's predominantly in countries now of, not so much in Latin America, that was 91, 92, but mostly in countries of Africa, the continent of Africa. So it's a global disease, and it's really associated predominantly with the environment. So the environment, let me just go back here. the environment really means the production with warming during the warm season of the year and the increase in the plankton populations, mostly along the coastal areas, and then the subsequent epidemics of the disease in countries like Bangladesh. Now, this is where we've done most of our work in the field. Students from the University of Maryland, my students, and I would traverse all the way to Bangladesh and work in the Bay of Bengal. This is the team from the University of Maryland back in 1975. This is Dr. Huck, who is now a research professor at the university in the Center for Infectious Disease here. So this was a team that went to Bangladesh, where, of course, water sources are crude, still in the remote villages, not so much in the major city. And everything goes on in the ponds. Unfortunately, the bacteria don't behave themselves and stay in one place. What we discovered was what we had learned in the Chesapeake Bay was exactly what was happening in Bangladesh. In other words, we couldn't always culture the bacteria, but we could pick them up in the water column using the staining technique. And we could, at different ponds, a pond, for example, that is used for religious purposes and not for swimming or drinking and so forth, or latrine, had very few of the bacteria. But the point that I would make is that the observations we had made in Chesapeake Bay held very well in Bangladesh. So we were able to perfect our model to say that, yes, there was person-to-person transmission, but fundamentally, the source of the bacteria is from the water column. Now, back to Bangladesh, the other observation we made was that in the Bay of Bengal, obviously along the coast, you could see the changes in water temperature. So we worked with NASA. This was 20 years ago. And we figured that if we could, at that time, satellites had gone up. It was the Landsat, the early one in the late 70s, early 80s, measuring chlorophyll, water temperature, and surface height, that is tidal factors. Now, these were all very useful for us studying cholera. And so what we found out was that by just a very simple calculation, we published this back in 2000, this is 19 years ago, we discovered that indeed, if you looked at the numbers of cholera cases in the lower parts of the Bay of Bengal over a couple of years period, and if you looked at the sea surface temperature and adjusted for 6 to 8 weeks, because first the water warms, the phytoplankton with chlorophyll capturing sunlight become very abundant, and then the zooplankton feed on the phytoplankton, they become abundant, and then the food source, phytoplankton has crashed, they crash, and then they break up, and Vibrios are released into the water column, and then you'll have cholera cases in Bangladesh where the villagers drink water without any treatment. And sure enough, this very crude correlation allowed us to say, look, we could measure and predict cholera by just using satellites. And the results were astounding in the sense that in Calcutta, now known as Kolkata, we could calculate that for every milligram per cubic meter of chlorophyll, there would be about 33% increase in the number of cases in Calcutta. And with a millimeter per day rainfall, it'd be about 7% increase in the numbers of cholera cases. And in Bangladesh, the same phenomenon. So it got to the point where we could actually use satellite because we had learned so much about the bacteria and the water system and their interactions that we could make these predictions with very great accuracy. So then we decided that we should go back to some of the early cases in India because it looked to us that there was always cholera along the coast, but there would be sudden epidemics, maybe every 20 years, that would occur inland. So we went back to the records at the University of London, where all of the data, all of the colonial records from the British occupation and the colonialization of India located. So by looking through the old records, and particularly around Delhi, where every 20 years there would be a massive cholera epidemic, and we were trying to figure out why that occurred in that fashion, but not along the coast. And we discovered that when we plotted the data that we transferred from these huge maps that are in the library in the museum in London to the computer, we found that what was a factor that was very important was temperature. When the temperature got above 30 degrees Celsius, then you would find large numbers of cases occurring. And similarly, relationship with flooding and rainfall. And back in 1990, a huge epidemic occurred in Latin America. And we were called in, our team from Maryland, because we had become experts at this point with the environmental aspects, to do the field work. And we worked also with the physicians in Latin America. And putting together all the data, we now were able to say, look, these are factors associated with these outbreaks of cholera. Rainfall, flooding, the level of the rivers, because the Gangetic Delta in Bangladesh is fed by the Himalaya, the mountain sources of water that come down in the spring with flooding and the monsoons, and then these patterns occur constantly. So the relationship is clear. And salinity, we had learned that from the work in the Chesapeake Bay, plays a major factor, because as it turns out, Vibrio cholerae requires sodium chloride to maintain its cellular integrity. You put it in distilled water, if you spin down the cells and then plunk them into distilled water, they lyse. Now this was observed by Robert Koch in 1890, that's a couple hundred years ago, but he called it the string test. That is, you would centrifuge crudely, as they used to do, sometimes with a handheld centrifuge, but you concentrate the cells, and then you'd add distilled water, and then you'd put an inoculating loop into that mass of cells, and as you drew it up, it was all sticky and gummy. Well, today we know it's DNA, because the cells have lysed. So this observation of salinity as a factor is an important one, but determined from our work here in the Chesapeake Bay. And coastal salinity plays a major role in terms of offshore distribution of plankton and therefore the bacteria. Now the assumption is that it was strictly fecal contamination, but Jim Caper, graduate student here at Maryland, now associate dean at the University of Maryland Medical School, Jim Caper was the one who discovered that in fact there was a negative correlation with fecal coliforms present. In other words, it's not simply a disease transmitted only by person to person, which it is, not only, but it can be. But the fact is the organism is naturally occurring in the environment. And in fact, a negative correlation for the parts of the bay where the fecal coliform count was high. So again, we improved the model so that we could say that quite clearly when the rainfall is well above average and there's a poor or damaged infrastructure for sanitation and for providing safe water, then there would be a very high risk of cholera. Now, could we have predicted the outbreak of cholera in Haiti? In fact, when you consider the various parameters associated with the outbreak in Haiti in 2010, there was a massive earthquake. Infrastructure that was not very good to start with, was severely damaged. What we found was that the postulation was that there was one source from which all of the cholera cases occurred. That presumed center of the origin, but at the same time that cases were occurring at the site of origin, they were occurring all over Haiti. Now, this is still a contentious issue, and I'm providing my views, which you may or may not agree with, but the point is that this was what was happening at that particular time. Now, this simultaneous occurrence of the outbreak was at a time when first in 2010, the earthquake had occurred in the early months, January, February, March. But that summer was the heaviest rainfall in 50 years, just at that, in August, September, October. And it was the hottest summer in 50 years. These were data we downloaded from the NASA records. And that combination was exactly as we had determined from all of the studies we'd done of the historic records in India, from the records in Bangladesh. And so it fit the model extremely well. And in fact, retrospectively, when the hurricane Matthew went around and bypassed, actually went over Haiti and on up the coast of the US, we applied our model and we predicted that, in fact, this would be the area with highest risk of cholera. And this is in 2015, a retrospective. And sure enough, this was the area just exactly where the cholera outbreaks were occurring. And we did a study as well in 2017, a couple years ago, of a study in Yemen. And we made our predictions of where the highest risk would be for cholera in 2017. And sure enough, that's exactly where the cholera occurrence was taking place. Now, this was picked, we wrote a paper, we published it in September of 2017. It was picked up by Scientific American. A little paragraph appeared about this model developed by the team at the University of Maryland in collaboration with West Virginia, which was my postdoc who had moved on to West Virginia. And we got a phone call in January of 2018 from the British Aid Agency. You know, if you could really predict cholera, as Fergus McBean, the Brit who called us, he said, could we work together? And in 2019, if you tell us where in Yemen we should be positioning our physicians and our medical supplies, we'll do that. We'll work together to see if we could actually reduce the outbreak of cholera in Yemen. And that's exactly what we did, and it worked out extremely well. In fact, we were able to predict, even at a greater scale, because the satellites that are now going up and that are up can measure a mile, a square mile. So we can actually measure temperatures of ponds, not just off the coast as we had to do with Bangladesh 20 years ago. And we were able to then predict just where in Yemen the supplies should be located, and the numbers of cholera cases went way, way down. Now we are working with the British Aid Agency and with UNICEF and with the Rockefeller Foundation, and we're now preparing to work in Africa as well as continuing our work in Yemen to predict when and where, how high the risk cholera will be, and be able to position supplies and use this as a predictive model. Now, the real-time prediction for Yemen, as I say, has been really successful, and we can measure 4 weeks in advance the highest risk, and we're working now to get 8 weeks in advance, and we're working with a data system from the British Meteorological Agency in England, where they have very powerful records of rainfall, temperatures, et cetera, in continents other than Europe and the US. So this is work underway and very exciting. Now, let me turn to another aspect of the aquatic system and climate change. And what does this have to do with Vibrios? Well, it's been a very interesting study. Different stations in the North Sea and off the coast of North America where sampling has been done. This is work done by the Southampton Marine Laboratory. About 40 years ago, they decided that they would carry out citizen science. They would give plankton nets to cruising captains, to ferry boat captains, to yachtsmen, to anybody who had a boat. They would give them these plankton samplers, and so these plankton collections go back 40 years. So working together with a team in Italy, Dr. Carla Pruso, a wonderful scientist, and a team at the Max Planck Institute in Germany, Southampton, and the University of Maryland, working together. What we did was go back to the plankton samples that had been stored and extracted the DNA, and then used molecular probes to determine quantitatively the numbers of Vibrio cholerae, Vibrio parahemolyticus, and Vibrio vulnificus. And so that was what we were able to do. We found that, particularly off the coast of North America, there was an increase, slow increase in parahemolyticus, Vibrio parahemolyticus, which is a causative agent. In fact, in the US, it's the major cause of food poisoning caused by eating fish that is not properly cooked. Now, what we were able to do is to link this increase that we observed with the last 40 years in the numbers of these potential pathogens and the actual cases that occurred up in Newfoundland, Maine, Massachusetts, and in Europe. And there was a direct correlation between the climate change, the warming of the water, the seawater, and the presence of Vibrios in the plankton that had been collected and stored over the last 40 years. This was published in the Proceedings of the National Academy of Science a couple of years ago. Now, one last example that I'd like to share with you, which is, again, even more exciting, I think, and that is using metagenomics to understand the populations of microorganisms and what we can learn about safe water using metagenomics. Obviously, this is the kind of development in molecular biology. I doubt that many of you even know about numerical taxonomy. That was what I did for my PhD thesis, and that was to write the first computer program to identify microorganisms. But it was really crude. It was an IBM 650. You had to wire it yourself. As graduate students got to use it in the attic of the chemistry building at midnight to 6 a.m., and you used punch cards, and God forbid you should drop the punch cards because you'd have to calculate it all over again. Anyway, what we did was code the phenotype, that is, whether the bacterium fermented glucose or broke down gelatin and so forth, and put all this on the computer and developed the first similarity indices. Now, of course, we use DNA-based pairs, but that was in the early days. And then here at the University of Maryland, working with Tiger, which doesn't exist now, it's the Venture Institute, but it was in Rockville, we sequenced, working with Mechelanos at Harvard, us at Maryland, and Tiger. We were the first to sequence Vibrio cholerae and to show that it has two chromosomes, a large chromosome and a small chromosome. And that's not a plasmid, too many housekeeping genes. When you cure it of the small chromosome, the organism cannot function, cannot reproduce. So we developed here at Maryland. I should point out that when I was director of the National Science Foundation, it was during 9/11, and that was there was an anthrax outbreak. I can talk about it now. In fact, it'll be in my book. But I was the chair of a classified committee that advised the CIA and the FBI on identifying the perpetrator of the anthrax incident. And we did succeed in identifying the source, but unfortunately, the perpetrator, assumed to be the perpetrator, committed suicide the day he was to be arrested. But we tracked it down by using sequencing and tracking the mutations that occurred. Essentially, in a very simplified statement, at Fort Detrick, there was a flask that was a composite of the anthrax grown up in Idaho and in Texas and at Columbus, Ohio, and at Fort Detrick to get a really strong test strain for the vaccine. But in the process of all these different fermentations, there were mutations. And so when you have a composite with all of the mutations, you got a really powerful evidence. And that was essentially how we did it. Well, that took 6 years. And so I decided, this is crazy. We have to do this much more quickly. So we developed a technique whereby we can extract from any sample, water, food, clinical sample, the DNA, and then with any sequencer, develop get the sequence. And then because we've spent a lot of time over the last 10 years building a really accurate database, 160,000 highly curated genomes of microorganisms. This allows us then to do a matching and binning of the K-mers and to identify all of the microorganisms. And then with a gene database, we can actually describe the presence of antibiotic resistance genes, pathogenicity genes, metabolism, etc. So this is very powerful. It allows us then to be able to observe the microorganisms of the gut, the skin, water, etc., and to combine it with metabolic pathways of the host, in this case human, and to be able to determine the interactions of the host and the microbial populations and the metabolic pathways and interactions. Now, as you all know, I'm not bringing anything new. There are good bacteria in our gut which regulate our innate immune system, synthesize vitamins that we can't synthesize ourselves, but also it is well known that there are many disorders now linked to the properties. Now, what we've been able to do is identify right down to species, strain, and substrain. That is very unique, and none of the other available algorithms are able to do that. Plus, we're able to do this in minutes and on a laptop and a server. We don't need a high-speed computer. So here at Maryland, we've developed a very elegant technique for identification. And why is strain important? Well, for Lactobacillus casei, one strain is used to make cheese, another strain is used to make Chardonnay, and a third is in the manufacture of yogurt. So strain is really important, especially in the case of E. coli. Strains we have in our gut are good to us. They produce vitamins, protect us against Clostridia, etc. But if you have an 0157H7 strain, that can be devastating with hemorrhagic enteritis. So let me give you an aquatic sample because, again, coming back to water. Water is critical. In this case, we've worked with a team at University of Texas, Dr. Chopra. A young woman built herself a zip line. She's on the zip line, it breaks, she falls into the water, gashes her leg on a rock in the water. They look for streptococcus because necrotizing cellular fasciitis, the flesh-eating bacteria, and this was essentially a case of flesh-eating bacterium, is usually streptococcus, but they didn't find any. Instead, what was isolated were 4 strains of Aeromonas, 3 that turned out to be clonal, that is, essentially identical, and then a very different strain. So working together, again, with another former student from the University of Maryland, Chris Grimm, who's out at FDA, works at the FDA labs out on River Road, and with Dr. Chopra in Texas and our team here at the University of Maryland, College Park, we were able to inject or do animal studies to find out just how this bacterium was operating. And when we injected the bacterium into mice, we found that only 1 strain went to the liver and to the spleen. This was kind of interesting because here was a mixture, but only 1 strain went through. Well, we knew this was going to be hard to get through to be published, so we engineered that 1 strain to be luminescent and kanamycin resistant and repeated the experiment. The only strain that went through was luminescent, kanamycin resistant. So we were able to publish this a couple years ago in PNAS, and we have a paper coming out in a couple of weeks, which describes the mechanism very elegantly of what's going on. In fact, the 1 strain that does not go through produces a powerful enzyme, very similar to that produced by Pseudomonas, that allows the other strain to migrate through to the liver and spleen. So here are 2 strains working together to create this devastating infection. And so dissemination requires the 2 strains to work together. Now, we've been looking at the gut flora of a variety of individuals, and a big study in India where cholera, again, is a major problem. And working with a cholera hospital, patients coming in the arrangement that we have with them is that they do all of the standard tests for bacteria, viruses, and parasites, send us the DNA, and then we do the analyses here using our technique. And we were able to find out some interesting things. First of all of the patients coming in with the symptoms of cholera don't carry Vibrio cholerae. Most of them do, let's say 80%, but 20% do not. So I think we have to redefine the disease as being caused by Vibrio cholerae, because in the cases that came into the hospital that did not carry the Vibrio cholerae, they were mostly a mixture of enteropathogenic and enterohemorrhagic E. coli. But the other thing that was fascinating was that we were able to discern the difference in the population, the gut population of the Indian versus Westerners, us in America or in Europe. And so this led to a very interesting study where not only could we identify the microorganisms that are present, but we also could determine that antibiotic resistance was much more common in the Indian population because antibiotics can be obtained from street peddlers. You don't need a prescription. And so they're much more indiscriminately used, hence the population of the controls are resistant or have the genes for resistance to a whole variety of antibiotics. And those coming in that we couldn't figure out what was going on had probably been treating themselves because the incidence was so much higher. So now back to water safety. This technique is being used very, very effectively because in California, as you all know, they're having severe fires and also drought. And Orange County, California, now uses sewage water. 50% of the water going into the drinking water plant comes directly from the sewage plant. The sludge goes out to sea. And then the other 50% comes from rivers, streams, and so forth. Is that water safe? So working with Orange County, California, we've been able to show that indeed the incoming, the gray area, these are all the kinds of bacteria that we've been able to identify as just a complete zoo of microorganisms. But after it goes through, let me just go back a minute, the treatment is the secondary effluent comes in, gets mixed with the good water coming in from rivers and so forth, treated with sodium hypochlorite, microfiltration, millipore filtration, reverse osmosis, irradiation, ozonation, water, chlorination, etc. So it's really well treated. So we found that the stuff coming in from the sewage plant has all kinds of microorganisms. But by the time it goes through the different steps, fewer and fewer microorganisms can be detected. And similarly with viruses. In fact, what we pick up by the time it goes through reverse osmosis are really only bacteriophages, which is really useful because then it confirms the microorganisms that we are detecting. But in any case, by the time it goes through reverse osmosis and then the final filtration, the only microorganisms that come through turn out to be iron bacteria, water bacteria that you would normally find in any tap water. And what's fascinating is that in addition, there's been a concern that with mixing sewage water with the natural river water, you're going to be transferring antibiotic resistance genes. And the bacteria coming out into drinking water would be highly resistant. That turns out not to be the case. We were able to show that, in fact, the water coming in has a lot of antibiotic resistance genes, but by the time it goes through millipore filtration, this is almost reduced by half, and by the time it goes through reverse osmosis, very, very few, and we find that the ultimate drinking water contains almost no antibiotic resistance genes. So this is very important because it means that wastewater can be very effectively used as a drinking water source. Now, again, it's important to be able to identify the strains as well as genus and species. And let me close with one last example. Again, water, which is the topic of the day. And that is, I've been describing to you using satellites, using DNA sequencing, doing a variety of elegant tests. What good can that be now for villagers in the remote parts of Bangladesh? Well, we concluded that if, in fact, most of the bacteria are resident on plankton, and we found that to be the case 30 years ago, right here in the Chesapeake Bay, that if we used a simple filter, a very simple filter, that is sari cloth, and if we folded it 4 or 5 times, and we did some electron microscopy here at the University of Maryland, and we found that it gave a nice 20 micrometer mesh, then these women could filter the water and thereby reduce cholera. And so we had an extension service, just like you guys here. They were the women that went out to the villages. We trained them, and then they went out to train the women in the villages how to treat the water by filtration. And it's simply a piece of sari cloth, folded about 4 to 5 times, put over the neck of the calash, pour the water through, and then bring the water up to the home to be used for drinking water. It was very obvious after filtration compared to things you could see swimming. It didn't take much more than one filtration procedure for the women in the villages to get it and to understand that this was important. And we were able to reduce cholera by 50%. So this is a technique that is very simple, but it's built on science. We went back 5 years later to see if they were still using filtration. The problem was that the control villages that we had not been introducing filtration had learned about filtration, so everybody was filtering. But it was good because we found out that we could pick up a herd effect. If families were filtering and you were too lazy to filter and you were in the middle of that group of families, you were protected because the herd effect provided filtration. These are some of the students that from the University of Maryland, colleagues from the International Center for Diarrheal Diseases Research, colleagues from all over the world who've been working with us. And especially I'd like to shout out for Anwar Haq. He and I have worked together for 40 years. Dr. Jutla, brilliant chap who's now at the University of Florida, who's been doing most of the monitoring. Hassan, Dr. Hassan, who's an adjunct professor, who is the informatics and DNA guy. And Sion Young, she's fantastic in building the library for us. And so water is really, really important. And the words of John Muir, the founder of the Sierra Club, when one tugs at a single thing in nature, he, and I inserted and she, find it hitched to the rest of the universe. Thank you.